Good morning, students. Today's our topic is photosynthesis. This is the first chapter of third unit. Third and second unit are plant physiologies unit. First chapter of third unit, photosynthesis. As you know, the father of plant physiology is Stephen Hales. फादर ऑफ प्लांट फिजियोलॉजी स्टीफन हेल्स. ये मैं आपको इसलिए बता रहा हूं क्योंकि इसकी हिस्ट्री में से एक क्वेश्चन बोर्ड के एग्जाम में आया हुआ है. तो उसमें द रोल ऑफ 5 साइंटिस्ट इन फोटोसिंथेसिस, तो एक साइंटिस्ट का नाम अपने को यही याद कर लेना है. ठीक है. व्हाट इज फोटोसिंथेसिस? कन्वर्टिंग इनऑर्गेनिक सब्सटेंसेस इनटू ऑर्गेनिक सब्सटेंसेस. एक डेफिनेशन होगी, converting inorganic substances into organic substances is called photosynthesis, and this capacity is found only in green plants. This capacity is found only in green plants. Next definition, process of making food. Process of making food in green plants is called photosynthesis. Means only green plants have the capacity to make their food, so they are called autotrophic. Third definition, look at the board. CO2 plus H2O is equal to organic acid plus oxygen. Here sunlight and chlorophyll. This is the full reaction of photosynthesis. Whenever we want to write a equal equation, then it is 6 CO2 plus 12 H2O is equal to chlorophyll, sunlight, C6H12O6 plus 6 H2O plus 6 O2. Now definition. What is the definition? According to Robenwich was the scientist who can give the definition in his own words. According to Robenwich, in photosynthesis there is reduction of CO2 and oxidation of H2O or chemically breakdown of H2O. By this, making of organic acid and oxygen is produced in byproduct is called photosynthesis. Or we can say in simpler words, what is photosynthesis? Photosynthesis is the process of making food with the help of carbon dioxide and H2O in the presence of light. Green plants make their food and oxygen produced as byproduct is called photosynthesis. Photosynthesis, the process by which the green plants use sunlight to make food from simpler molecules and carbon dioxide and water. Photosynthesis can also be described as the process by which green plants synthesize organic substances from carbon dioxide and water using energy absorbed from sunlight by chlorophyll. So who can absorb the sunlight? Chlorophyll are the source which can absorb the sunlight and by which sunlight is converted into chemical energy. Or we can say one definition of photosynthesis is that what? Physical energy or sun energy is converted into chemical energy. Now, the equation can be summarized. The raw material are carbon dioxide and water is absorbed from the air and water from the soil. Next, the energy needed for chemical reaction is derived from light absorbed by chlorophyll. Third, simple sugar like glucose are produced and oxygen is byproduct of photosynthesis. Chloroplast contain a green pigment chlorophyll, which is the site of photosynthesis. Now, next topic is our history of photosynthesis. In history, we have to learn only the role of scientist in photosynthesis. First thing, you remember it, whenever we provide light to a plant continuously, then that plant will be dead. अगर हम किसी प्लांट को कंटिन्यू लाइट देते हैं, मीन्स अपन जानते हैं कि नेचुरल कंडीशन में दिन में क्या होता है, एट द डे टाइम प्लांट ऑब्टेन सन एनर्जी एंड इन द नाइट it cannot obtain sun energy. So in the night plant can make organic acid and in the day time they produce oxygen. So regular interval of light and dark is necessary for photosynthesis, and photosynthesis reaction is completed in two steps: light reaction and dark reaction. Dark reaction further divided into three parts: C3, C4 and CAM cycle. So whenever we want to memorize or we want to write down history or the role of scientist in photosynthesis, then each reaction will be given by other scientists. So their role we can also write if we not remember these scientists. मैंने बोर्ड पे कुछ साइंटिस्टों के नाम लिखे, उनके रोल है वो देखिए. सबसे पहले जीन सिनेबियर ने बताया कि फोटोसिंथेसिस की रिएक्शन में CO2 का रिडक्शन होता है. तो द रोल ऑफ CO2 इन फोटोसिंथेसिस टेल्स बाय जेन सिनेबियर. नाउ द नेसेसिटी ऑफ वाटर इन फोटोसिंथेसिस विच कैन एब्जॉर्ब्ड बाय सोइल टेल्स डी सेसर. Chlorophyll needed in photosynthesis tells by Durochet. Sunlight is required in photosynthesis says that engine house, and in photosynthesis reaction organic acid or a simpler glucose is produced state by engine house. इंजन हाउस ने क्या बताया कि फोटोसिंथेसिस की रिएक्शन में सनलाइट की नेसेसिटी होती है, और इंजन हाउस ने क्या बताया कि ऑर्गेनिक एसिड का फॉर्मेशन होता है, और इन्वायरमेंट जो है वो प्योर होता है फोटोसिंथेसिस की रिएक्शन से. किसने बताया? प्रिस्टलेन. ठीक है, ये अपने साइंटिस्ट का रोल हो गया. और मैं आपको एक अगर आंकड़ा बताऊं तो क्या होता है कि फोटोसिंथेसिस में 170 million tons of dry matter annually performed. Among them 90% of that was produced by oceans. मीन्स जितना फोटोसिंथेसिस होता है अपनी अर्थ के ऊपर, उनमें से 90% बिकॉज ऑफ, आप लोग जानते हैं 70% अर्थ पर क्या है? ओशियन है. और टोटल फोटोसिंथेसिस का 90% जो है वो ओशियंस में होता है, और 10% जो है वो टेरेस्ट्रियल प्लांट फोटोसिंथेसिस करते हैं. तो ये अपना हो गया हिस्ट्री. नाउ नेक्स्ट टॉपिक इज साइट ऑफ फोटोसिंथेसिस. साइट ऑफ फोटोसिंथेसिस, 1 mark question. Answer is chloroplast. And the structure of chloroplast you study earlier in junior, we further study it here. क्लोरोप्लास्ट क्या है? फोटोसिंथेसिस की साइट है, यानी फोटोसिंथेसिस वेयर हैपन्स, तो दैट इज हैपन्स इन द क्लोरोप्लास्ट. और ईच सेल ऑफ ग्रीन प्लांट्स और मे बी लीफ आर द ग्रीन, तो ईच सेल ऑफ लीफ कंटेन 20 to 40 क्लोरोप्लास्ट इन इट. मीन्स ये मान लीजिए सेल है, ये वैक्यूल है, ये क्या अपने क्लोरोप्लास्ट है. ठीक है, तो एक सेल में 20 to 40 क्लोरोप्लास्ट आर प्रेजेंट. और 1 millimeter square of leaf contains 50,000 of क्लोरोप्लास्ट. ये भी एक क्वेश्चन 1 mark का बनता है, तो आप लोग ध्यान में रखना. 1 millimeter स्क्वायर में 50,000 क्लोरोप्लास्ट प्रेजेंट होते हैं. अब यहां पे अपना क्वेश्चन बनता है स्ट्रक्चर ऑफ क्लोरोप्लास्ट. सो लुक एट First of all, we can categorize all the cell organelles in three parts. Which are the three parts? Double membrane organelles, single membrane organelles, and membraneless organelles. So chloroplast is the double membrane organelle, and two organs of the cells are called semi-autonomous organ. Which organs are called semi-autonomous organ? Chloroplast and mitochondria are the semi-autonomous organelle of the cell. Why these organs are called semi-autonomous organs? Because they have their own DNA in it. So they are called semi-autonomous organelle. Then it is plastidial DNA. Plastidial DNA means क्लोरोप्लास्ट और डीएनए में क्लोरोप्लास्ट और माइटोकॉन्ड्रिया में क्या होता है? इनका ओवन डीएनए होता है. सो दे आर कॉल्ड सेमी ऑटोनोमस ऑर्गन. देन क्लोरोप्लास्ट इज ए डबल मेम्ब्रेन स्ट्रक्चर. इफ वी वांट टू लुक एट द structure of leaf, then there is two epidermis, upper epidermis and lower epidermis. Among these epidermis, this tissue is called mesophyll tissue. Mesophyll tissue can be further divided into two parts in pellicid and spongy. Then the cells of pellicid tissue and spongy tissue contains क्लोरोप्लास्ट. दिस इज पेलिसेड टिश्यू एंड दिस इज स्पंजी टिश्यू. तो इस स्पंजी टिश्यू में यहां और ये पेलिसेड टिश्यू में यहां ये क्या है? क्लोरोप्लास्ट है. एंड क्लोरोप्लास्ट इज ए डबल मेम्ब्रेन स्ट्रक्चर. फर्स्ट आउटर एंड सेकंड इनर. द स्पेस बिटवीन two membrane also present there, which is larger than the thickness of outer and inner membrane, and this space is called plastidial space, or we can say interplastidial space या intermembrane space. Outer membrane and inner membrane. Chloroplast has two main structure in it. The matrix of chloroplast, or we can say the liquid part of the chloroplast, is called stroma. Remember students, there is two reaction of photosynthesis, light reaction and dark reaction, and the dark reaction of photosynthesis is happens in stroma. ये 1 मार्क का क्वेश्चन पूछा जा सकता है कि पहले अपन ने बात की क्या, साइट ऑफ फोटोसिंथेसिस इज क्लोरोप्लास्ट एंड लाइट रिएक्शन की साइट क्या है, तो वो है अपनी ग्रेना, एंड डार्क रिएक्शन की साइट क्या है, स्ट्रोमा. ठीक है, स्ट्रोमा इज द लिक्विड और मैट्रिक्स पार्ट ऑफ द क्लोरोप्लास्ट. अमंग देम देयर इज अ सॉलिड पार्ट प्रेजेंट इन क्लोरोप्लास्ट इज कॉल्ड grana, and grana is like a collection of coins. Whenever we arrange coins one upon one, then the structure perform that is called grana. So this is called grana, and there is so many grana, and this grana is the site of light reaction. Grana, and whole granas are called granum, and each part of granum is called grana, and grana is the site of light reaction, and it is a solid structure just like coins collection. There is sometimes two grana are combined with the membrane, that is called thylakoid membrane, or we can say stroma lamella. Stroma lamella. Among the stroma lamella, the particular unit which can absorb the light, among stroma lamella or a thylakoid, there is some drops or some micro bodies are present, which is called quantasome. And quantasome has Remember, quantasome has 230 chlorophyll molecule. I can write down on the board. Quantasomes has 230 chlorophyll molecules. Among them 160 are chlorophyll A and 70 is chlorophyll B. 48 carotenoids, 46 quinone, 116 phospholipid and some enzymes are present which are useful in reaction of photosynthesis. So what is quantasome? Quantasome is the unit of photosynthesis. Quantasome is the unit of photosynthesis which are present between grana or thylakoid membrane, which has 230 chlorophyll molecules. Among them 160 chlorophyll A, 70 chlorophyll B, 48 carotenoids, 46 quinone, 116 phospholipid and some other enzymes. Now we are discussing here photosynthesis. So you know, in the presence of light, plants make their food. So light has three quality which can we discuss later, but here among that light 400 nm to 700 nm nanometer light is useful in photosynthesis, which is start from violet to red. Among them sunlight only 400 nanometer to 700 nanometer light was useful in photosynthesis. So this is called PAR, photosynthetic active radiations. Photosynthesis active radiations is the light in between plants makes their food. Our next topic is photosynthetic pigment. Photosynthetic pigment means the source which can absorb the light are called photosynthetic pigment. And another thing you know that what is pigment? Pigment is the source of color in the any living organism. Here three pigments are present in plants. First chlorophyll, second carotenoids and third phycobilins. Chlorophyll, first of all, chlorophyll was discovered by Will-Stotter, Stoll and Fisher. Chlorophyll was discovered by Will-Stotter, Stoll and Fisher. There is 7 chlorophyll molecules are present in the plants, which are chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E and bacteriochlorophyll and bacterioviridin. Means 5 chlorophyll A, B, C, D, E and bacteriochlorophyll and bacterioviridin are the chlorophyll molecules present in the plants. Among them, the molecular formula of chlorophyll A is C55H72O5N4Mg and chlorophyll B C55H70O6N4 and Mg. There sometimes examiner can also ask the structure of chlorophyll molecule. So you have to remember the structure of chlorophyll molecule has two parts. First, a tetrapyrrole ring or porphyrin ring, or we can say for your better understanding, the structure of chlorophyll is just like a racket of badminton. Among them, in racket of badminton, the head part of racket, or we can say the head part of chlorophyll is made of porphyrin ring or tetrapyrrole ring, and the handle of the racket or handle of the chlorophyll molecule is of hydrocarbons tail called phytol. The photosynthesis ring is 4 smaller pyrrole rings in which each small ring there is a nitrogen atoms. All 4 rings bind with the metal atom magnesium present at the center. Just like in the center there is magnesium and nitrogen is surrounding this magnesium, and here 4 tetrapyrrole rings are performed and a long tail is performed by methyl groups which is called phytol ring. remember, here I can write in chlorophyll A there is on the head methyl group is present, but in chlorophyll B aldehyde CHO group is present. Next pigment is carotenoids. Carotenoids and phycobilins are the accessory pigment for photosynthesis. The light which can't be absorbed by chlorophyll, that will be absorbed by carotenoids and phycobilins, and this light energy transferred to chlorophyll. That can be studied in the process of photosynthesis. Carotenoids are discovered by Vikander in carrot. Generally they are red and of two types, carotenes and xanthophyll. The molecular formula of carotenoid is carotene is C40 and H56. xanthophyls में क्या होता है कि यही मोलिकुलर फॉर्मूला है C40 एंड H56, बट ऑक्सीजन और प्रेजेंट होती है. जनरली केरोटीन जो है वो रेड कलर के होते हैं. एग्जाम्पल लाइकोपिन, अल्फा केरोटीन, बीटा केरोटीन. नेक्स्ट xanthophyll molecular formula C40 H56 O2. They are generally yellow or brown in color. Example lutein, zeaxanthin, cryptoxanthin, neoxanthin. यहां एक नंबर का क्वेश्चन फिर बनता है, which are the carotenoids, which carotenes are present in all plants? Here we can write some examples, but there is two pigments, one carotene and one xanthophyll is present in all the green plants. That are, you have to remember, beta carotene and lutein are the pigment which are present in all green plants, and other examples present in some other plants. Last, phycobilins. Phycobilins are prefer the color of the plants, mainly red and blue. The blue phycobilins are present in blue green algae, or we can say cyanobacteria. Cyanobacteria में क्या होता है, फाइकोसाइनिन जो कि ब्लू कलर शो करता है, और फाइकोइरिथ्रिन रेड कलर शो करता है जो कि रोडोफाइसी एल्गी में प्रेजेंट होता है. तो ये आज का अपना टॉपिक. कल अपन पढ़ेंगे शेष भाग इस अध्याय का.